crossing the road on your own or someone else holds your hand and helps you to cross the road. Students, what am I talking about? This is regarding simple and facilitated diffusion. Very basic yet very important mechanism of transport across cell membrane. In this video, you are going to learn little bit overview about diffusion and comparing and contrasting features of simple and facilitated diffusion. So come on, let's watch this video. But before that, do not forget to subscribe my channel Homeostasis 99. So come on, let's watch it. So students, let's learn first what the diffusion is. So diffusion is basically the movement of substance from its higher concentration to lower concentration. Why the movement happens from higher to lower concentration? Because towards higher concentration side, the kinetic energy is high of that particular substance as compared to the lower concentration side. So here in this we can see that suppose this is a chamber which is filled with some gas, and these are the gas molecules which are colliding with each other. They are moving here and there, and they are having high kinetic energy here as compared to the outside of this box. So because of this higher kinetic energy, collision happens and the substance will move from out of the box, from the box to the outside environment. So because of this, we say that diffusion is the movement of substance from its higher concentration to lower concentration, because where the concentration high, energy will be high. The substance will move from high energy side to low energy side. But that doesn't mean that substance is not moving from lower to higher. It is also moving from lower to higher, but the movement is less as compared to this side motion. Okay, so this is the difference. That's why we say it net diffusion. So in this diagram you will understand it better. Like in this container, we are having some solutions which are separated with the help of a membrane, and this solution A, the side A as higher concentration solution, and this side B is having lower concentration of solution. So here the solute is high, and this solute particles will move from A to B because the energy is high here. They are also moving from B to A, but energy is low, right? So the movement, you can see here is a big arrow is shown. Here the size of the arrow is less. So magnitude of transfer of the substance from energy low to energy high is less as compared to higher concentration to lower concentration. It means from A to B the substance is moving more, and B to A it is moving less. So here इन दोनों का जो difference आएगा उसको आप बोलोगे net diffusion. ये नेट डिफ्यूजन ही है जो आपके मेम्ब्रेन के अक्रॉस काम करता है, बिकॉज डिफ्यूजन इज अ मेथड, वेरी इंपॉर्टेंट मेथड ऑफ ट्रांसपोर्टेशन अक्रॉस द सेल मेम्ब्रेन, ट्रांसपोर्टेशन ऑफ वेरियस सब्सटेंसेस अक्रॉस द सेल मेम्ब्रेन. तो ये आपका सेल मेम्ब्रेन है विच इज कंपोज्ड ऑफ लिपिड बाइलेयर, राइट, एंड द सब्सटेंस हैज टू क्रॉस दिस मेम्ब्रेन. Okay, so crossing of this membrane can happen with the help of diffusion. Okay, simply from its higher concentration side to low concentration side, and this is called as net diffusion because this is the net magnitude of the movement of the substance across the cell membrane. There are various factors affecting it which are defined as Fick's law of diffusion. बिकॉज मेम्ब्रेन के अगर दोनों तरफ सब्सटेंस ट्रांसपोर्ट होने वाला है तो बहुत सारे फैक्टर्स हैं जो उसको अफेक्ट कर सकते हैं, जिनको फिक्स लॉ के साथ आप एक्सप्लेन कर सकते हो. एग्जाम में बहुत बार ये पूछा जाता है, एक्सप्लेन द फैक्टर्स अफेक्टिंग रेट ऑफ डिफ्यूजन विद द हेल्प ऑफ फिक्स लॉ. सो फिक्स लॉ स्टेट्स दैट J मीन्स नेट रेट ऑफ डिफ्यूजन इक्वल्स टू D, that is the diffusion coefficient, A is the surface area, T is the thickness of the membrane, and concentration gradient, that is suppose your concentration is moving, your substance is moving from ECF to ICF, so concentration outside is high because it is moving through diffusion, outside it is high, inside it is low. So difference between concentration outside minus concentration inside will be your concentration gradient. Okay, so relationship क्या है? Net rate of diffusion is directly proportional to the surface area of the membrane. जितना बड़ा surface area होगा substance को move कराने के लिए उतना ही ज़्यादा diffusion रहेगा. Thickness, membrane का thickness जितना ज़्यादा होगा, suppose if there is any inflammation happening across a membrane, and because of that inflammation some fluid is gathered here and the swelling of the membrane happens and some damage happens, so this is increasing its thickness. So if the thickness is increasing, the net rate of diffusion will be less. Concentration gradient, जितना ज़्यादा surface के दोनों तरफ concentration का difference रहेगा उतना ही ज़्यादा diffusion का rate रहेगा. D is the coefficient, diffusion coefficient. It depends upon the permeability of the membrane for that particular substance and the molecular weight, size of the molecule, type of the molecule, molecular weight. उन सबको मिला के एक constant बनता है जिसको आप बोलते हो diffusion coefficient. So these are the factors affecting net rate of diffusion according to Fick's law. So now we'll discuss the types of diffusion. That is a main part of our today's video. Types of diffusion is simple diffusion and facilitated diffusion. This diagram. In this diagram, this membrane lipid bilayer is shown, which has some protein molecules embedded in it. So that is a lipoprotein bilayer. And here you can see that three types of movements shown. One which is directly happening through and through the lipid bilayer. It is crossing easily the lipid bilayer. Okay. Second type, this is your first type of movement. Second type of movement is happening through a tunnel, which is not into the exposure of this lipid molecules, but here these are the protein molecules which are making a passage for a particular substance to pass through it. This is called the channel transport, okay, or transport through channel proteins. This is your second type of transport shown. The third type of transport shown in this diagram is what? The substance is moving, but here these are some binding sites shown. Means the substance has to attach on these binding sites and then it will be transported on any of these sites. So this is called the carrier protein. Carrier protein has the binding sites on it. Okay. So when the substance binds to it, the conformational change happens and then the substance will move from one side to another side. Okay. This is your third type of transport. So these first and second are included in simple diffusion, where the substance is moving on its own across the membrane, either through lipid bilayer or through protein channel. No other extra support is required. Only through its concentration gradient it moves. This is called simple diffusion. And in facilitated diffusion, someone has to hold its hand, the substance's hand, and it has to help crossing the road for that. And in facilitated diffusion, the substance has to bind on this molecule and then the conformational change happens and then the substance has to be transported. So this is called facilitated, means the substance is moving though because of its concentration gradient, but it needs some extra support of a protein molecule, right? So these are the types of diffusion: simple diffusion and facilitated diffusion. Now what are the differences in these? Because exam mein question aayega, define diffusion and give the types of it and compare and contrast between these types, or directly they can ask compare and contrast simple diffusion with facilitated diffusion, because both of these are the transporting mechanisms across the cell membrane that we have. Now we are moving towards comparing and contrasting features between the simple and facilitated diffusion. how they are similar and how they are different from each other. So first of all, the comparing features. Direction of transport. In simple diffusion, direction of transport is towards the concentration gradient, means from higher concentration to lower concentration, right? And in facilitated diffusion, the same, from higher concentration to lower concentration, because both are the diffusion. From higher to lower concentration, the substance is going to move. External use of energy, not required, because substance is moving through its own kinetic energy. Here also it is not required. That's why we call these types of transport as the passive transport. This is a passive mode of transport across the cell membrane because it does not require external source of energy. The substance will move on its own with its kinetic energy. Lipid solubility. Lipid solubility is present here in case of simple diffusion if the substance is crossing the lipid bilayer. So lipid soluble substance can cross directly the lipid bilayer like oxygen, carbon dioxide, alcohol. These are lipid soluble substances, can directly cross the substance. In simple diffusion, it happens for lipid insoluble substances also, but that is required the protein channel. Through this protein channel, the substance is transported. So what are these substances? Like ions, sodium, potassium, calcium, okay, chloride. So water, these are the substances which are lipid insoluble, transported through the protein channels. In case of facilitated diffusion, the substance is having lipid insolubility. Substances are not likely soluble in lipid, so they need some help with the binding of this carrier molecule, and then it will be thrown on other side of the membrane. Substrate specificity. Substrate specificity, if here in case of simple diffusion, if the protein channel is utilized, this protein channel is specific for specific substance. Like we have sodium channels on our membrane, we have potassium channels, we have chloride channels, we have calcium channels. There are different channels for different substances. So substrate specificity is here in case of channel transport, not in case of lipid soluble substances which are transported directly through the lipid bilayer. In facilitated diffusion, the substrate specificity, yes, it is present because substance needs to bind on its binding site before it is transported. So this binding needs the compatibility, and compatibility gives the substrate specificity. So specific carrier molecule for specific substance. So these are the features which are comparable between these two. Now the contrasting features. The molecular size. Molecular size, the substances or the molecules which are going to be transported through simple diffusion, these are small molecules, smaller as compared to facilitated diffusion where the large molecules are transported comparatively. Larger molecules like glucose, amino acids. Such substances are transported through facilitated diffusion. These are the larger molecules as compared to the simple diffusion molecules which are smaller like gas particles, oxygen, carbon dioxide, water, ions, all are smaller molecules. Next feature, Fick's law of diffusion. Fick's law of diffusion, as we already studied it, what it says, the rate of diffusion is equal to dA upon T into concentration outside minus concentration inside, which is the concentration gradient, right? So here in case of simple diffusion, this Fick's law is followed as it is. Here in case of facilitated diffusion, the Fick's law is followed up to some extent if we specifically talk about this effect of concentration gradient. Refer this graph. What this graph says, this graph gives you the relationship between the concentration gradient and the rate of diffusion on y-axis, here on x-axis the concentration gradient. So on x-axis, as you increase the concentration gradient, there is increase in the rate of diffusion. This yellow graph, this graph gives you this information कि आप जैसे जैसे concentration gradient बढ़ा रहे हो, आपका rate of diffusion बढ़ रहा है. This is the feature of simple diffusion, okay, as we discussed. Now, in case of facilitated diffusion, if the substance is transported through a carrier molecule, here the concentration gradient is rising, there is rise in the rate of diffusion. After that, as we further increase the concentration gradient, there is no rise in the rate of diffusion. The rate of diffusion becomes constant here. So this is called V max, the maximum attainable volume, maximum attainable speed of diffusion as you increase the concentration gradient. That is called V max. So V max is the feature of facilitated diffusion. Now why this is happening? Why it is not increasing further? The reason is that here on this carrier molecule you have the binding sites, okay, and on which these substances are going to come and bind it. Then the configurational change will happen and then this substance will be thrown this side. But after it throws, it will go back to its initial condition. So this whole thing will take some time. So if we increase the concentration gradient as much as that it will occupy all the carrier molecules and all the binding sites, then what will happen, if you further increase the concentration gradient, there is nothing remain for them to bind with because all the sites are occupied. The V max, the maximum achievable speed of diffusion as you increase the concentration gradient, and after that the speed of diffusion is not increased as you further increase the concentration gradient. So V max is the feature of facilitated diffusion. Now we are moving again to our table, this comparing and contrasting features. So Fick's law of diffusion. So Fick's law of diffusion is followed completely here and partially in case of facilitated diffusion. Vmax is not present in case of simple diffusion. Vmax is present in case of facilitated diffusion, that we already seen. Saturation kinetics. Saturation kinetics is the phenomena that we discussed already, that when the all binding sites, all the carrier molecules are saturated by their substrate molecules, there is no more binding site available for new molecules. So saturation point comes in case of facilitated diffusion and there is no saturation point in case of simple diffusion. भाई simple diffusion तो आपका या तो लिपिड बाइलेयर के थ्रू होगा या प्रोटीन चैनल के थ्रू होगा. सो ये दोनों ही अगर रास्ते खुले हैं तो ये आराम से जितना भी कॉन्सेंट्रेशन ग्रेडियंट है उसके हिसाब से सब्सटेंस ट्रांसपोर्ट होता रहेगा. द नेक्स्ट पॉइंट इज योर stereospecificity. Stereospecificity means the molecule, if sometimes the molecules have two types of isomers which are called L isomer and D isomer. L stands for levo and D stands for dextro. So in which direction it rotates the polarized light, according to which the L and D isomers are there of a single molecule, which are the mirror images in their structure. So for example, we have L glucose and D glucose, right? And stereospecificity means the substance is binding with any particular type of isomer, not with the other. So for glucose carrier molecule, we call it as GLUT, glucose transporter. So glucose transporters, we have around 14 families known of GLUT proteins. These glucose transporters or the GLUT proteins are specific for transporting D glucose, which is normally occurring glucose in our body, in living tissues. So, and it is not able to bind with L-glucose. So it has stereospecificity. It is binding with the isomer D but not with the isomer L. So stereospecificity is present in case of facilitated diffusion, whereas stereospecificity is not present in case of simple diffusion, because it is not something like the substance has to bind somewhere. It is just going to cross the membrane. So for that, the stereospecificity doesn't matter in this case of simple diffusion. Now coming to the next feature, which comes with again the same property of binding with substrate, which is the competitive inhibition. Competitive inhibition means any substance which is similar in structure can bind with that in replacement of that particular substance. Like if we talk about glucose, on glucose receptors on the GLUT proteins, galactose also can bind. So these two substances are competing with each other to bind on this transport each other. So competitive inhibition is present in case of facilitated diffusion, and there is no binding with something, so no competitive inhibition present here in case of simple diffusion. So this is all about the comparing and contrasting features of simple and facilitated diffusion. So these all points you can discuss when you discuss your answer. So students, I hope I have made myself clear enough. If you like this video, press the like button. If you want me to prepare video on some other topics, let me know about that also. And any query regarding this topic, then you can send me messages and share it with your friends, those who need it. Bye-bye.